And Petrolac, it's my pleasure to introduce Dr. Mark Rubin, who is a professor and director of the Department of Biomedical Research and Bern Center for the Precision Medicine at the University of Bern in Switzerland. Previous to his move to Europe 6 years ago, Dr. Rubin was a group leader at Weill Cornell Medicine Institution that remained engaged to the prostate spore as a project leader. Dr. Rubin is a recognized world-renowned leader in prostate cancer genomics and pathology and in precision medicine. Dr. Rubin's laboratory led a series of landmark studies defining distinct molecular features of prostate cancer, revealing pathways that are perturbed and drive different types of this cancer. Furthermore, he has translated many of the genomic discoveries into clinical tests that are currently patented and standardly used in the diagnosis and treatment of prostate cancer. He founded the Engler Institute for Precision Medicine and most recently the Bern Center for Precision Medicine. Dr. Rubin has published around 300 manuscripts, including those in major top journals. Today, he will tell us about his latest work in the minor spliceosome exploring novel cancer vulnerabilities. Dr. Rubin.
Well, it's great to.
Dr. Herbst may want to say something.
Okay, sure. Herb.
No, I thought Katie would want to say something, Dan.
We're okay. So everybody's okay. Okay, great. So it's great to be here. And I think last time I was here is definitely over 10 years ago. So it's great to come back and visit. And hopefully I can figure out how to move the slides forward on this. Okay. There we go. So just the following mostly non-relevant disclosures. A lot of people asked in 2017 when I went to Switzerland, what I was going to do and why I was going. And so you heard that there are some initiatives that I've been involved in. So setting up the Bern Center for Precision Medicine and running a new department and reorganizing a department of biomedical research. But one of the decisions I made when I moved was that instead of taking projects that I had, I would start from scratch and really start new projects. And just, I see we have a lot of trainees in the audience, and I just think it was a very, I think it's been an exciting time. I didn't account for the pandemic, but it's certainly been an exciting time to think about new projects, what you think is important, and actually going in places that feel a bit uncomfortable scientifically, just because you need to learn new things. And so I think I left all the projects with this prostate spore and we started new projects. So I'll tell you a little bit about two things that we worked on more recently. Just as way of background, so many of you are familiar with prostate cancer, but I'll just as a means of background, just to remind you that the landscape for prostate cancer therapy has really changed dramatically. If we go back 10 years ago, or 2010, even more than 10 years ago, it was relatively androgen deprivation therapy based with taxanes, but it's very different than the landscape today. And I'm not expert oncologist, I'm a pathologist by training, and I'm not going to go into all the details, but just to point out that there are many choices. And if you go to clinical meetings and listen about, what's going on, just all the time, there are new opportunities. The one thing I would point out is that as patients are being treated with anti-androgen, various types of more potent anti-androgen therapies, such as enzalutamide and abiraterone, resistance is obviously inevitable. So patients can do well for a short time, or they can do well for a long time. Eventually they'll fail. But there are other opportunities now. There's PARP inhibitors, so that's quite exciting. Obviously, there's immunotherapy for a small subset of patients where it seems to be effective. And then there's PSMA treatment, so imaging and treatment. And these are, again, really more therapies. There's problems with all of them, but there is a lot more opportunity now for treating advanced prostate cancer. Resistance remains a major focus of our lab's work and other labs now to try to understand why patients are failing. And what I think is very important is to think about in all of these settings, what is the key clinical question? At what time should we change therapies or introduce new therapies? So that's one of the main things we're focusing on. So I'm going to make a few comments about lineage plasticity and the resistance framework. I'm going to talk a little bit about some work that may be relevant to the lung spore in the context of switch SNF and epigenetic modulation. And then I'm going to talk about a new story that's unpublished that has connections with both Yale and Connecticut. It's on minor splicing. So my main goal today will be hopefully to tell you about something that you may not know a lot about, and maybe we'll all learn something together about minor splicing, and maybe you'll have some insights as well, and that we can talk about them. So just a few comments about resistance and lineage plasticity. I've been showing these slides for a few years, and I think it helps me a lot at giving a framework for thinking about what we do clinically in the setting that in the context that most of the types of therapies are very index oncogenic pathway driven. So if you have estrogen receptor, if you have HER2, if you have EGFR as a main pathway that's activated, BRAF, et cetera, the index oncogenic output is being targeted. And so then you have resistance related to that, but there are alternate effectors and there are also alternate states that can occur. So in the context of prostate cancer, the androgen receptor is the main oncogenic target for most of the therapy, whether it's androgen deprivation therapy or targeted ligand-targeted therapy or therapy based on decreasing synthesis of androgens or hormones. So you'd expect, and it is the case, that most of the mutations or resistance occur in the androgen receptor. So there's amplifications, mutations, and so this is what you expect. But what we also are seeing emerging more and more frequently with very potent anti-androgen therapy are alternate pathways. And this is getting into the theme of lineage plasticity. So if you switch from an adenocarcinoma to something else, you're no longer driven, so you're AR negative. And what are the mechanisms then that lead to this resistance is what we're trying to understand, and a number of other groups are doing the same in prostate. Now, obviously, this is very interesting in other cancers like bladder, lung, and breast. So just to be specific, as a pathologist, we look under the microscope and we see this is an adenocarcinoma. It's very pink. The cells have a lot of cytoplasm. And after anti-androgen therapy, a certain subset of these patients, and what I'm showing here is just the types of therapies that patients may receive, very strong, potent anti-androgen therapy. But after these therapies, where most patients, almost all patients respond initially, many of the patients will have resistance, and there are many different flavors of resistance. The most common, so I don't want to mislead you, the most common is probably still something that looks like an adenocarcinoma. It still can be responsive to more potent anti-androgens, but a subset, maybe 10 to 15%, will have something that looks like this. So it's a neuroendocrine prostate cancer that's no longer responding to AR therapy. And something, usually these patients have very, very aggressive disease. If you look under the microscope and perform immunohistochemistry, you'll see that you have the adenocarcinomas are very positive for androgen receptor expression and are not positive for neuroendocrine markers such as synaptophysin. And for neuroendocrine cancers, you lose AR protein expression. Now that's important, just one important point, and this is probably true in many other cancers. If you block AR and now you have a neuroendocrine tumor, the pathways for AR, so you might see at the transcript level an attempts for androgen receptor to produce protein, but protein levels are usually very low. So you don't see active androgen receptor protein. These patients have a very poor outcome. So patients who are now diagnosed with neuroendocrine prostate cancer clinically or by biopsy usually have anywhere from 7 to 12 month median survival. So very poor, and there are very few therapy options. In showing this slide, I can show that we have an example of adenocarcinoma all the way to small cell cancer. You can see in the adenocarcinoma, it's positive for PSA, which is a surrogate for androgen signaling, and that it's negative for neuroendocrine markers here. And then as the tumor seems to, as we see different tumors where you have this neuroendocrine tumor, we see a positivity for the neuroendocrine markers. Now, I think the important point here is an additional comment is that this is all from the same patient. This patient had metastatic prostate cancer, and these are from almost the same lesion in different areas. And so it's possible that you not only have this trans differentiation or lineage plasticity, but it may be in the context of mixed histology. So I think it's important to just remember that heterogeneity is obviously is an important component of resistance. A few years ago, probably around 2013, we started a Stand Up to Cancer, PCF funded, Prostate Cancer Foundation funded trial, which was one of the first precision oncology trials. There are a number of PIs leading this, Charles Sawyers, Arul Shanayan, Levi Garraway, among others. Phil Kantoff. In this study, one of the things that as pathologists was great is that we're able to look at now over 1,000 cases of patients that were prospectively collected who were failing androgen deprivation therapy in one sort or the other from multiple institutions. And we could ask the question as a pathologist, if we used AR signaling as one score to look how adenocarcinoma tumor was, and neuroendocrine signaling, because we had transcriptomic data as to how neuroendocrine the tumor was, we might expect to see a division into two camps. And we could expect that there might be a third gradient of what tumors that are in transition. This is the real data. So it doesn't look like I think this was at the time we had around 500 cases, but many, many of the cases are sitting here. So high androgen receptor signaling, only some of them. So I mentioned about 15%, 10 to 15% of our cases showed neuroendocrine features. What was surprising, and this is really the first prospective study that I was aware of this or have been involved in prostate cancer in this setting, was that when we looked in this quadrant here, where we expect to see these very blue cells and neuroendocrine cells that I showed you, we see things that don't quite look like neuroendocrine prostate cancer. They look like something else. So it has a squamous appearance. Take my word for it if you're not a pathologist, but it doesn't look like a typical neuroendocrine tumor. And then in an area where we see very strong expression for the androgen receptor signaling, not just androgen receptor, but signaling, we see something that looks like a neuroendocrine cancer. These were not mixed up. We confirmed everything. So I think what is very important is that the histology, so the phenotype and the genotype don't necessarily correlate that well. And this causes obviously a lot of confusion for classification, which is very important for treatment. This is a case that's in the middle, and it shows a very unusual histology. So for somebody doing prostate pathology for many years now, we usually see certain morphologies, but we don't see these odd-looking nuclei. And I think this is really related to a few factors, but at least one factor is that patients are getting very potent antiandrogens. Another factor is that they're living longer, so they're able to survive longer. So we're seeing changes that we previously hadn't seen. So I think this is part of the natural evolution of the cancers, just that maybe we haven't seen this. And then in prostate, maybe in other cancers as well, metastatic biopsies are not the norm. So usually we're not actually seeing what's going on as you treat patients. So this was eye-opening for me as a pathologist. When we looked at the molecular alterations in some of these cases, they tended to have P53 and RB alterations in these cases, as well as a neuroendocrine. I'll talk a little bit more about that. But this leaves us with a picture where we have a spectrum of disease. It's very difficult just looking under the microscope to classify it. So it really suggested to us that we need to come up with other ways of classifying it. And I would say right now, we don't have that. I mean, we're moving towards that, but we don't really have that established to date. As I just mentioned, the phenotype and the scores or the signaling scores that you have from transcriptomics don't match perfectly. So, and we also recognize they're probably intermediate states. So I think those are important observations. So we start out with a model where we wanted to look at very discrete differences, adenocarcinoma and neuroendocrine tumors. In fact, when we look more carefully, what we see is a whole collection of intermediate morphologies, genotype, and also transcriptomic and probably epigenetic alterations. About the same time, we wanted to do a very focused study at looking at these extremes to ask the question, what are some of the other factors that might play a role? So genomics, but also epigenetics and transcriptomics were applied then to a set of, I think at the time, 50 or 60 cases where we had bona fide neuroendocrine cancers diagnosed by pathology and adenocarcinomas, and we compared them. And I'm not going to go through these published studies, but I just want to highlight, I think the important finding for us was that we expected to see some really exciting genomic alteration that would characterize the difference, but we didn't. What we really see is a commonality that RB1 and P53 are very commonly altered in the small cell phenotype, less commonly altered in the adenocarcinoma, but we do see it. And that in general, the overlap in genomic alterations, copy number alterations is very similar, except for, let's say, the androgen receptor, where that would be something that's very different and altered in adenocarcinoma, but not in the small cell. When we looked at the data in combination, so looking at DNA, RNA, and epigenetics, and then asking the question, which element best explain the differences between the two phenotypes, I think importantly, the numbers may differ depending on the configuration of the population, but certainly epigenetics or methylation helped explain the difference quite well in this population. And I'm not going to say too much more about that, but we think methylation or epigenetic alterations are very important. So here's a figure that we made for a review, and the concept was that at some point, adenocarcinomas, there's an inflection point where adenocarcinomas maybe go through some sort of stem-like state and then undergo lineage plasticity. Now, I think for people in the lung cancer field, this was something not unexpected, but this is something we wanted to explore in prostate as well, because we think that at some point, these around 10 to 15% of the cases are no longer responding to AR-driven therapies and become AR negative. In a sense that then they will then either transdifferentiate to small cell or AR negative, neuroendocrine negative. So another type of AR negative phenotype. And I think that was supported by some of the morphologies we saw from the trial. I list a number of studies. I'll just very briefly make a few comments about the polycomb gene, so EZH2, and also the switch SNF work that we've done. But I think others are quite interested also for the lung cancer spore. It's probably quite interesting thinking about epigenetic regulation and where we stand today. So I think this is a very nice review that came out after two papers were published in Science, suggesting that there's a stem-like state that occurs before you go to neuroendocrine or to AR negative state, and that one of the key players is probably EZH2, so a polycomb gene that's responsible for repression of a large number of genes associated with AR signaling. And this is just one key experiment from one of the papers from David Goodrich's group, where they demonstrate that if you knock out in a mouse model P53 and RB, and that's what's shown here, the tumors are no longer sensitive to the potent anti-androgen enzalutamide. And so you can see that the tumors are now continuing to grow, even in the presence of enzalutamide. If you use EZH2, you can reactivate or resensitize these tumors to the epigenetic drugs that are used now clinically as EZH2 inhibitors, and you can reactivate AR sensitivity. So this is just a concept that this is a stem-like state that's potentially reversible, and I think that's an interesting concept. I'm happy to discuss more about those. There's some controversy a little bit about that, but I'll just leave it at that for now. We were interested in the flip side of the polycomb complex, which is a repressive complex, looking at the switch SNF complex. And the things that really were of particular interest to us were, one, that there's an embryonic switch SNF complex that goes to other types of complexes. So you can have a stem-like complex, but also a neuronal complex. And the idea was that in development, it's very important that you're able to have neural development, and switch SNF complex activates and can facilitate a neural development and playing an important role at H3K27 marks of acylation. So this is important in development, but in cancer, it's also known that the switch SNF, particularly the ATPases, SMARCA2 and SMARCA4, are often altered. And of particular interest in many of the synthetic lethal screens, SMARCA4 and SMARCA2 come up as sort of key findings. And so there's been a great interest in translationally developing drugs that would knock out SMARCA2 specifically in SMARCA4 altered tumors. And unfortunately, as many of us are aware in knowing what's happened, it's very difficult to actually have a specific SMARCA2 inhibitor. So most of, whether it's an ATPase inhibitor or a PROTAC, this has been it's been challenging. So most of the work is really focused on knocking out both and creating a synthetic lethality. What we found in sort of as is very common in prostate. So prostate cancer research tends to look at things differently because for whatever reason, prostate is a little bit different than other cancers. And we were interested in looking at the overexpression of SMARCA4, which is not usually the case in many of the tumors that have lost SMARCA4 expression. So these are complicated complexes, and I'll simplify it by just saying that there's a working component, which is ATPase, and there are two paralogs, SMARCA4 and SMARCA2. Unfortunately, they also have different names. So there's Brahma and BERG1, but I'll just call it SMARCA4 and SMARCA2. And the idea is that most of the work is going and targeting these. And we were interested to see what happens in prostate cancer progression, whether there were mutations or alterations in these, in the ATPases. What we found is that when we look, as you look in prostate cancer progression, we tend to see an increase in SMARCA4 and a decrease in SMARCA2. So that's a little bit different than what's seen in some other cancers. But the other important feature is as you look at prostate cancer progression, we also see an increase in some of the neural components of the switch SNF complex that are associated with the neural complex, which is called BAF53B as one of the proteins. So just to visualize this as a pathologist, their core complexes don't change. So going from localized disease or benign tissue to advanced disease that's neuroendocrine positive, you don't see any differences. But what you do see is the neural marker shown here, BAF53B, is only expressed in their endocrine tumor, as you'd expect. And what we see here is that SMARCA4, so one of the two ATPases, the paralogs, is very highly expressed in the neuroendocrine tumors, or maybe the stem-like types tumors that we see. SMARCA2 is not expressed. So we were originally thinking that for us, it would be very interesting to modulate SMARCA4 in the context of prostate cancer. And just as an aside, we don't see any mutations that are seen in other cancers in either of the paralogs or in any of the SWI/SNF complex members. When we look at pathology again, if you look at cases where you have adenocarcinoma here, and then you have some neuroendocrine cancer in the exact same tumor, you can very nicely see some of the things I'm telling you about. So the small cell expression of synaptophysin is seen here. The BAF53B, which again is a neural component, neural protein component of the SWI/SNF complex, is expressed there, but not in the adenocarcinoma, so that's important. And then here is, I think, a very important finding, at least in the published paper, where we took an organoid that was a neuroendocrine organoid from a patient and looked at it. Now, we're seeing heterogeneity, so that's one thing that's important, is even in a patient-derived organoid that's been passaged many times, we see heterogeneity. And what's interesting here is SMARCA4 is expressed here, SOX2, so a transcription factor that's very much involved in stemness, is expressed, but the neural markers that I mentioned, BAF53B, are not expressed here, but they are expressed in the SMARCA2 area. So it looks like the paralogs play a different role, and modulating them may do something very different. And so we think that there's a dynamic activity going on. You have some cells that are more poised to be neuroendocrine and others that are potentially that are potentially still very stem-like and potentially reversible. David Goodrich, who in collaborations has shared some of the organoids that he's developed through some of these mouse models. So I mentioned that he's developed mouse models where he's done knockout of P53 and RB. They've also knocked out P10. And so in black, we have P10 knocked out. In blue, we have both RB and P10, and in red, he included P53 knockout in these various mouse models. And you can see that in the mouse models, you also have the same observation that SMARCA4 goes up when you knock out RB1, and a little bit of the controversy is whether you need P53 or not, because we don't see much change when you add P53, but that's a sort of another discussion. And then SMARCA2 goes down dramatically when you knock out either P53 alone, RB alone, or with P53. There's some other interesting findings. The methyl transferases increase when you do this, and I'm not going to talk about it today, but there's, I think, an interesting story related to the epigenetic side of methylation that also occurs in this setting. One other, I think, important point is in this mouse model, we then did proteomics on the patient-derived organoids from this model. And what's interesting and what you'd expect is if you have these models where you knock it out, that we see overexpression of SOX2, which I showed you in the human samples. We also have SMARCA4s overexpressed. But what's quite interesting is that if you take this model out of the mouse and now put it and just grow it in vitro, I should say in vitro, not in vivo, what ends up happening is you don't see these changes anymore. So just one other comment, that environment we think is very important. And I think when a lot of us, when we're doing our organoid experiments, we have to be mindful that the results could be very different. And there's, I think, more to come about this. I think Wouter Kartoff, who's was at Memorial, now is in Lausanne, and others are working very much in this area. So just final comments about switch SNF. So we were sort of surprised at the end of 21, so beginning of 22, to see a paper from Roel Schneid's group, because we've been following this field for a while, and we've known about the toxicity for the PROTACs if you try to target switch SNF. And they have a very nice paper where they developed a PROTAC that was not toxic. So they showed a lot of very nice toxicology data from in their paper that's not toxic in at least the mouse models that they show. And that the switch SNF PROTACs for combined SMARCA4 and SMARCA2 are exquisitely potent against AR-sensitive prostate cancer. And so we found that quite interesting. We have been following up. We had actually been working on something similar and had come to a similar conclusion, but extended a little bit, and just point out that our collaborators, Yu Chen and Ekta Khurana, who was formerly a trainee of Mark Gerstein, is here, had a very nice paper in Science where they used a TAC-seq, so an epigenetic approach to classify prostate cancer. And I won't go into all the details, but again, essentially, in addition to AR-sensitive advanced prostate cancer, they also came up with the Wnt signaling pathway, a stem-like, and neuroendocrine. So I think it's a good working classification for these advanced cancers, more than just AR negative, but AR negative could be stem-like, it could be Wnt signaling or neuroendocrine. I'm sure this will change, but I think it's a very nice study. So when we applied this classification and used a PROTAC that we had acquired in collaboration with Genentech that was recently published for lung cancer, a very nice study. We were also able to demonstrate exquisite sensitivity to AR positive, but also to some of the other subclasses, so extending it beyond AR sensitivity. So we think that these are very useful approaches. Unfortunately, the toxicity, at least in our hands, is very high. And so I think the strategy now is really to try to come up with more specific either SMARCA2 inhibitors But for prostate, the question is, should we also be looking for a SMARCA4 inhibitor? And so that's something that's of great interest. So just to summarize this part of the presentation, I wanted to just give you sort of the landscape of what's going on in prostate cancer and with regards to resistance. There's genomic and epigenetic players, which I mentioned, RB and P53 being two of the main players, which are probably necessary but not sufficient. And then there's very interesting stories developing in epigenetic regulation. I didn't talk about REST for the first part. I'll talk about REST, which is an inhibitor of neural differentiation. I'll talk about that in the second part. And then I think that this is now emerging where, based on these different alterations, there may be different subclasses. So I just want to highlight, we've modified a little bit a review that we had a few years ago, where I think microenvironment plays an important role, and there are different pathways that are taken. Are they unidirectional? Are they reversible? I think that's going to be a very important translational issue. And I think for those treating patients, obviously wanting to identify the time point where people will best respond to novel therapies will be important. So that's one thing I wanted to tell you about this morning. And now I want to tell you about something that's entirely new to our group. And when I go for bike rides around my house, I don't know if there are a lot of bike riders. I know David Grimm might be listening, and he's a bike rider. But when you are biking and you see that there's a slope of 27 degrees, that's really steep. I mean, so in Tour de France, 20 is starting to become extremely challenging. I've been in races where I can't go up 20. I'm walking. So when I see this, I'm always thinking one day I'm going to go down and try going up this, but I haven't reached that day yet that I'm going to try that. Maybe I'll have to wait for an e-bike, but I'm not there yet. But so I think in our lab, we'd like to take on challenges. And when Anke Augsbach, who's a postdoc in my lab, who had been working on the Switch SNF project, said what she really wants to work on is splicing. And she said, I'm particularly interested in minor splicing. I had a problem, which was one, I knew nothing about minor splicing, and I had no idea whether this was really a good use of her time. So I think this is always an important point for a PI to decide, okay, are we really going to take this on? So I learned a lot, and I'm going to tell you about what we learned. It's a work in progress, but I think it potentially is an exciting new area for us. And helping me with my process of education was Rahul Kanadia, who's close by. So he's just up the road at UConn, and his postdoc, Kyle Drake, who very helpful. And this is also a collaboration with Mark Gerstein's lab here at Yale. So there's a very strong Connecticut component. So what I did know about splicing was I knew from Gunnar Retsch's group at the TCGA that splicing is occurring very often in cancer, that it can lead to neoepitopes. So I was more familiar with aberrant splicing in the context of potentially immunotherapy. What I was also aware is that it plays a potentially important role in the regulation. So this is a hallmark-like figure showing all the different areas where splicing can play an important role. But I didn't know anything about minor splicing, so I had to learn from Anke, Rahul, and others. And so I knew that when genes create messenger RNA, but in order to do that, you have to take the exons and somehow the introns have to be spliced out to go from a pre-mRNA to an mRNA that can be translated. And so the question is, how does that actually occur? And I'm not expert, but I have learned that the vast majority of introns are excised through the major spliceosome, which is U2 spliceosome. And in this spliceosome, there are small nuclear RNAs that recognize consensus sequences, both at the 3' and the 5', but also branching points, and allow for this splicing to occur. So that's the vast majority of all proteins. And it turns out, and this is what Anke was interested in because of work she had done in her PhD, that there's a minor spliceosome which recognizes introns that have a different consensus sequence. And it's also referred to as the U12 spliceosome. And so this is something that's really very understudied. In homeostasis, I'll tell you a little bit about what we think it does, or what it's believed to do, but it's very important in cutting out minor introns that are part of genes that also have major introns. So I'll show you what that means in a moment. So the idea is that in a typical gene that has a minor intron, which we're going to refer to as minor intron gene or MIG, these genes have one minor intron, usually not more than one, and that it requires a specific machinery to cut this minor intron out. Now, I just will focus for most of the presentation on one element of the minor spliceosome, which is called U6 attack, which is one of the catalytic components of the minor spliceosome. And that in homeostasis, it's believed that under stress situations, the conditions are such that U6 attack is not degraded, and it allows for minor splicing to occur. So whatever this gene is, it might be important in a stress situation. It allows now MAP kinase signaling occurs and allows for the excision of this minor intron mRNA to be produced, and then translation of whatever that protein will be. So that's an important component. This is an evolutionary conserved event. So minor splicing is not only in humans, but also throughout the evolution, minor splicing has been maintained, and there's some interesting exceptions. The main role of minor splicing is seen in development. So Rahul, who's our collaborator, is really an expert in neural development. And so in patients who have germline alterations or other alterations in minor splicing, they see developmental problems. And there are many developmental diseases that are described or attributed to errors in minor splicing. For cancer, there's only two really known diseases, so Peutz-Jeghers and myelodysplastic syndrome, that are associated with minor splicing alterations. And in total, there are around 750, maybe 800 genes that have a minor intron, which we can refer to as minor intron genes. So Anka asked some questions that I think are pretty straightforward questions to ask in the beginning of this project. That is, do we see any evidence of minor splicing alterations in cancer progression? If so, is it preferentially during disease progression? So is it something that you see more so in advanced or resistant disease? Is this an active, functionally active event? And that would be very important if we're going to attribute this as a key causative role or playing a part in the cause of cancer progression or resistance. And what happens if you inhibit this? And can you reverse some of these features? And so I'll show you some of the work we have. It's as I mentioned, this is a work in progress, but we do have a first paper in revision, which seems like it's been in revision for, I think, almost a year now, but hopefully we're getting closer to that point. And so a first study was in silico. So one of our collaborators for this project looked computationally at protein-protein interactions, taking 26 of the most well-described prostate cancer genes and asked the question, what is the direct protein-protein interaction with these minor intron-containing genes? And as you can see on the right, some very interesting genes that just highlight BRCA, for example, here, or kinase A, MYC, which are genes that are very much associated with prostate, but also other cancers, have as a very close relationship, direct interactions with minor intron containing genes. So that's sort of a first interesting hint. I'm going to tell you about minor splicing in disease progression, but before I just want to emphasize why I'm going to focus on U6 attack. I mentioned that it's a catalytic component. So if you have a gene that has a minor intron, somehow the machinery comes together and has to cut out this minor intron. And the reason why U6 attack we think is very important is because it's a dynamic component of this process, where it's really like the last step. So it has to come together with U4 attack, and this catalytic subunit now cuts out the intron. And that's why it's probably very carefully regulated in homeostasis. So that's the reason why we're focusing on U6 attack. And the idea is, as I mentioned, that in stress, we think that U6 attack is used to help remove the minor intron for a subset of genes that are probably important in responding to stress. So that's our hypothesis. So what happens if you look in prostate cancer this is probably also true for other cancers, but we focus on prostate cancer using some of the common cell lines and patient-derived organoids and arranging them going from benign all the way to neuroendocrine disease, so trying to cover the spectrum. We see an increase in U6 attack expression. As I mentioned, there are other components of the minor spliceosome. They also show similar overexpression. We were able to then show this in situ using RNA-ish, and we're able to show that you can see higher expression of U6 attack, but also other components as you look at primary prostate cancers, but also higher in primary prostate cancers that go on to metastasize, and then in metastases even higher. What's important is this actually functionally doing anything? And so what Anka did was she used two vector systems that are designed so they have either one intron, and the intron is either a minor or a major intron. And with this luciferase assay, she's able to demonstrate expression if it's working. So if it's working as a minor intron excising machinery, or is there a major intron excising machinery working? And I think importantly, when she looked at the major intron splicing activity, it remained fairly similar throughout all the different types of model systems she looked at, but only highly expressed for minor intron splicing in the advanced cancers, which is intriguing, suggesting that the activity is increased. This is a very simple vector system, and we've since developed more complex vector systems that I could tell you about. At the transcript level, we see, and these are just some of the cell lines in prostate cancer, we see very high expression of U6 attack, but also other minor splicing components. But then also the MIGs, the minor intron-containing genes, are more highly expressed, so as we might expect. Now, Mark Gerstein's lab helped us with, I think, an important part, which is also extending this to other cancers. And the question really is, are these MIGs that we see that are alternatively expressed in cancer, are they potentially very useful in distinguishing different cancer types? So we would hypothesize that the MIGs are expressed, but probably different in different tissue types, just like we see in progression. And so Mark's group was able to create the silhouette plot here, looking at 23 different tumor types from a pan-cancer analysis. And they were able to show that in this experiment where you go from 0% MIGs, and then, so this is a dilution experiment, all the way to 100%. So this is done by doing many, many iterations. You can demonstrate that the best model is a model where you include the MIG. So it shows that they have a very strong ability to distinguish different cancer types. But specifically for prostate, when we ask the question, if we look at benign prostate tissue from GTX database versus localized prostate cancer or advanced prostate cancer, we see that the MIG genes do a very nice job without in any selection of a subclass of the MIGs to distinguish the different groups. I think it's important because it suggests, I think, that these genes, for whatever reason, for evolutionary reasons, are important in stress, but they also are probably reactivated or useful for cancer progression. And as I mentioned in the first part of the presentation, it's very important when we think about resistance to think about probably two types of resistance. One is still related to AR signaling active tumors, and we have to find ways to attack them, but also AR negative tumors. And I mentioned that there are these four categories. So the question really is, how can we gain any insight into that? Well, one of the things that Anka had read about and was known is that MAP kinase signaling is very important for U6 attack stability. And so she asked a question using an antibiotic that stimulates MAP kinase signaling, and isomycin, and was able to demonstrate that when you activate MAP kinase signaling or JAK-STAT signaling, you see only increased expression of minor splicing in the neuroendocrine tumors, which is sort of interesting, not in the AR sensitive tumors. When she knocked it down, it was the same thing. So I think that's important. And then in a separate study looking at the effect of androgen stimulation, she was able to demonstrate in different model systems, whether it's a LINCAP cell line, which is very androgen sensitive, or a LINCAP cell line that overexpresses AR, she's able to demonstrate that minor intron activity is increased when you increase AR. And can be modulated through that, but not major intron splicing. So basically, I'm trying to think about how A6 attack could be stabilized or modulated. AR signaling could play a role, but also MAP kinase signaling. And so she started developing a framework of how to think about this, thinking that neuroendocrine tumors might require MAP kinase signaling to stabilize U6 attack and allow for minor intron containing genes to be expressed, whereas AR sensitive tumors might do it in a different way through AR signaling. And we have more data that help support this. One thing that was nice is there were two high impact papers that came out talking about a subclass of prostate cancer that is really driven by JAK-STAT. So this is quite interesting. And these are stem-like tumors, which would fit in nicely with the model of minor splicing being driven by a MAP kinase signaling. So in just the last part, now I want to go into what happens if we try to target U6 attack specifically. And we hypothesize that this will have a direct effect on the ability to excise these introns that are minor introns. And in fact, that's the case. So when Anka used small interfering RNAs to knock down U6 attack, she demonstrated using a minor splicing index, which looks for missplicing. So now the introns are no longer being spliced out, and so the data is analyzed. And it consistently shows in all the different model systems that if you knock down U6 attack, it functionally has the effect of not allowing these minor intron containing genes to excise out the introns. For the experts in splicing, I won't go into that. I'm not an expert in splicing, but cryptic splice site alterations seem to be the most common, but here you can actually determine the specific types of splice sites. So we have experts here, but I'm not an expert to talk about that. Anka performed transcriptomics and proteomics, and in the context of knocking down U6 attack, the main finding, I think, are that in the different cell lines, we saw different genes that are altered, which I think goes into the view that this is going to be very context specific, the regulation of minor splicing. There were certain themes that emerged, and I think the important theme that I just would highlight would be two. So one would be cell cycle, and also DNA repair were two themes that came out when we look at the common genes that are altered in these different model systems. And so here's an example of an analysis where it shows some of the common themes in cell cycle, as well as DNA alterations came out as being altered when you knock down U6 attack. In single cell sequencing, FACS analysis, I'm not showing all the data, just to highlight that we see a G1 arrest when you knock down U6 attack, supporting the view that minor splicing plays an important role in cell cycle. In a series of experiments where she looked at both cancer cells, so this is a androgen receptor sensitive, a castration resistant tumor that is still probably sensitive to AR signaling, and you knock down U6 attack, you see a decrease in growth of tumor cells. We see no change when you look at either mouse fibroblasts or human fibroblasts. And in co-cultures, one of the reviewers, I think, is a good point. Do we see preferential changes occurring in co-culture? And we actually see only the tumor cells are affected by U6 attack knockdown. This is probably the key therapeutic or translational slide, which is that in AR-sensitive tumors, if you treat with enzalutamide or knock down U6 attack, you see basically the same result, which is a decrease in confluence of cells. So this is using IncuCyte. If you then go to cell lines that are AR resistant, and there's just 2 cell lines, but we've done it or more, you see that the tumors are no longer sensitive to enzalutamide or the anti-androgen therapy, but continue to be very sensitive to knocking down U6 attack, which is a promising first step for thinking about, does this have any therapeutic translation? Although many, many steps away from actually having therapeutic translation. We extended this to the Memorial Sloan Kettering and Cornell patient-derived organoids that represent a range of AR positive and AR negative prostate cancers. And we see the same effect that you can, by knocking down U6 attack, you can decrease cell viability and confluence. I'll skip the videos, but we can also see this in benign prostate cell lines that we have, as well as the cancer cell lines. I think the movie, okay. So the very last piece I just want to bring up is an interesting concept. And this is something we call poison peptides. Maybe somebody's used this in another context, and maybe it's not the right context, but right now we're just, working title is poison peptides. And the idea is that when the minor intron splicing occurs, it forms a protein that it potentially plays an important role in cell cycle and helps stabilize the cancer cells. But when it's not excised, the question is what's happening with these message, and is it becoming is it just undergoing degradation, or are there some sort of aberrant or other types of isoforms that are formed? And just want to show you an example that we were quite intrigued with, which is related to REST. So REST is a transcription factor, plays an important role in neural fate regulation in development. And so most of us know about REST. When REST expression is present in cancer types, we know that it prevents neural differentiation. And when it's down, we expect that you may see neural differentiation. And this is in cancer, we see this occurring quite often. There's also a isoform that's known, but little known, and that's called REST4. And REST4 forms a dimer with REST1 and prevents it from binding to DNA, and therefore it allows neural differentiation. So if REST4 is present, you do have neural differentiation. And this is just showing it in a slightly different way. So there's a small there's a small mini intron here, a small exon here that needs to be excised in order for you to go from REST4 to REST1. So what I wanted to show you is that then Anka asks the question, well, what does she see as far as REST expression? As expected, it's very low in neuroendocrine prostate cancers. This is from our stand-up cancer data. But she sees higher expression of REST4 in these cases. So that's something we had never looked at, because we never thought about looking at the different isoforms. And if you think about the endogenous expression of REST4 in the different cell lines, we also see the same thing. So very low REST4 expression in the neuroendocrine tumors. When she knocks down U6 attack, she increases REST expression. But I think what's really interesting is if you look at the protein level and you look at the different isoforms, she's able to demonstrate in neuroendocrine models that if you knock down U6 attack, and again, this is not perfect, but it's a beginning of developing a hypothesis that she sees a decrease in REST4. So at least a hypothesis is developing that we can think that in the normal state of these advanced prostate cancers that are neuroendocrine, they have high levels of REST4, which prevent REST from being functional and allow for neuroendocrine maintenance or differentiation. And that knocking down U6 attack, but clearly other ways of modulating REST, could also lead to a situation where you knock down REST4 and you increase REST. Anyway, so I'll leave you with just a nice image of Switzerland, so a nice lake that we like to go hiking around. And just a summary, so I've told you about minor splicing, how it's really a very small component of the spliceosome, and that we think U6 attack is quite interesting because it plays an important catalytic role and potentially represents an important opportunity for therapy. And most important slide is just to make sure I acknowledge all our great collaborators. So in addition to Anka, we have the Rubin Lab members, as well as members from Rahul Kanadi's group at University of Connecticut and Mark Gerstein's group here at Yale. They've been really, really helpful in letting us think about this in really broad terms. Thank you very much for your attention today. Happy to take any questions. Okay.
So, yeah, so the floor is open.
I don't know
Are
if
there
you need to-
questions for Mark? I can get started with a question. That was really nice. Thank you very much, and wonderful to hear about both of the stories on the SWI/SNF complex and on the minor intron splicing. So one of the questions that I have, and it's something that's something that we think about a lot as well, is the sensitivity of tumors to SMARCA4 modulation, for example. And one of the things that you mentioned was, or it seems like these PROTACs seem to be most effective in tumors that are AR sensitive too, which is kind of interesting if we think about SMARCA4 perhaps exerting its functions in the resistant tumors. And I was wondering if you have any thoughts on sort of that paradox,
Well, yeah,
if it is a paradox.
Yeah, I'll just expand a little bit. So I think the things, just to reiterate what I said though also, and you're well aware of, is that if you do any synthetic lethal screens, SMARCA4 and SMARCA2 come out as always winners. They're always there, right? And so I think that the PROTACs in effectively, hitting both regardless, are, great targets. I think so the question is, why are the AR sensitive tumors so, why are they exquisitely sensitive to this? We think that that's not the case. So we actually think that it extends beyond, it's not just AR. And the complexity we have is that we think that SMARCA4 is something you want to decrease. But we know that if we do that, SMARCA2 has to be maintained. So there's a problem with the PROTACs because they're extremely toxic. And so how to get around that. The other thing also, I'm sure you're aware, and you've maybe seen this, is that when you use the PROTACs, you can actually get nice resistance very quickly. And as you expect, where would the resistance be? It's in VHL or in one of the genes associated with how the PROTACs are designed. So we have nice experiments I didn't show, but where we've, can cycle through resistance, which is going to be different than the ATPase type resistance. So in a rules paper, I think it was exciting to see that you can potentially target cancers. I think there's a bit of skepticism in the issue of toxicity, because I think the field, and in Bob Yauk's paper from Genentech, I think they nicely described that the toxicity is a major issue. And so since a specificity, whether it's for SMARCA2, but we think it would be interesting to have SMARCA4 specificity for prostate. So I don't know. I mean, but we don't have either at this point. So.
Thank you. Yes. So yes, please go ahead.
You might have to repeat the question for me because I didn't hear it. Okay. Do you think you could?
I think it would be hard. Would you like to come up?
So I think you have to say it in the microphone so people can hear it who are
Right,
listening.
I think so.
And also I can't hear very well. So,
I think we'll.
so this is,
Thank you.
so what's your name?
My name's Krupa.
Oh, nice to meet you. So
Nice to
we have a
meet you.
guest speaker here.
My question was about SMARCA4's the sensitivity with the PROTACs in the prostate cancer cells. I'm wondering if you think that the SMARC-A4, like the BRG catalytic subunit is active within a Y-SNF chromatin remodeler enzyme complex in that context where it is being affected, or if you think it has a separate activity. And I'm wondering, because you mentioned EZH2 as well, I know can have polycomb independent functions where it associates with the androgen receptor, if something similar might be going on.
Yeah, so that's a great question. So, and the way that I didn't show data, but the way that I think we've been going at this has been to look at also our ATPase inhibitors. So we also have data for the ATPase inhibitors. The PROTACs are more effective, but they're more toxic. And so, but inhibiting the ATPase does, make it have a similar effect. But again, it's not specific to SMARCA4, SMARCA2. So it's hard to dissociate that. And we have SI data, we have CRISPR data for knocking out what happens if you knock out SMARCA4 and SMARCA2. So that's one problem. The other problem that we have, which we didn't discuss at all, is that it's really difficult to chip these proteins. Some people can do it. So Sigalkotic's group is world expert in that. We haven't been able to achieve that, unfortunately.
Other questions? Yes. The role of progesterones
Okay,
as well
so-
as androgens.
Estrogen.
Estrogen,
Okay,
estrogen.
so that's one of my favorite topics. I love that topic. So the role of estrogen in advanced prostate cancer. So I think it seems like it's paradoxical, like it shouldn't be that important, but estrogen receptor and androgen receptor, they have very similar binding sites. So there are a lot of half binding sites that are regulated. So I think it's an important role. And that it's also known that other nuclear hormones are activated in the context of AR depletion. So we think it's important. And in prior studies, we'd seen it's part of progression, but we didn't specifically focus on that here. So it's, yeah.
In plasticity, its role in plasticity
So the role of estrogen in plasticity
or progesterone.
or progesterone. So I think in stemness. So I think Charles Sawyer's group has looked at that, and I think there's the view that it does play a role in resistance and is seen associated with stem-like state, but maybe not neuroendocrine state. So, yeah.
Thank you, Joe.
One quick question from one of the two medical oncologists here. Thank you for coming out with a great talk and sharing that with us on the biology of the neuroendocrine prostate cancer. So my question is actually about the therapies for neuroendocrine prostate cancer. As you know, these disease is in a way sort of sensitive to sun-based chemotherapy, and you show that you're the and other alterations. I wonder if there are any genetic alterations that sort of predict response to sun-based chemotherapies or any, gene-denting disease. I wonder if you can comment on this.
Well, I'm a pathologist, so I better not comment on clinical oncology therapy. But the only thing I would say is that there's great interest in, epigenetic regulation, which are toxic. And, a close friend of mine, Johan de Bono, I was just with him the other day in Basel, and we were talking about this. So I think there are a number of studies that are coming down the line, but it may not be, so I presented these different categories. It may be for AR negative, but not neuroendocrine tumors, that there are, some new therapeutic targets. HER3 is a target. There are other targets that are coming up, some common to lung cancer as well, but no winners yet, no successful winners. And then hopefully I point out in the first part of the presentation how difficult it is to actually classify these. So once it's AR negative, I think there's still complexity. So we're far behind breast cancer and maybe lung cancer as far as being able to accurately classify what needs to be treated. So I think that in all the in the advisory boards, I think the common conclusion after 2 or 3 days of discussions are always the same, which is that we just need to treat everybody and see what works, which is sort of depressing, but that's sort of where we are, I think.
I think we have similar issues in lung cancer and resistant tumors, trying to
Okay.
figure it out.
But again, I'm a pathologist, so that's just my personal opinion, not don't take that beyond that.
We have one last question in the back.
Is it that there will be any role of differentiate so like the cluster not as
I'm sorry, I can't hear.
Do you think there's a role for differentiation therapies in prostate cancer, kind of analogous to the retinoic acid
Yeah.
in AML?
No, that would be, so, I mean, so one thing, I just maybe not directly related to your question, but being able to model differentiation would be phenomenal. So if we had model systems where it could show, I know like in AML and other cancer and other hematopoietic cancers, you can show differentiation or blocking differentiation. And I think we don't have those models. I think the closest thing we have right now is the develop of like the stem-like state. And then, ideally we'd like to flux back between adenocarcinoma. So we're starting to see some of the organoid models in mouse, but we don't really have that ability yet. So I do think what you're saying is interesting. And my view would be that if you could tell which tumors are going to transition, you'd want to treat them as early as possible with the other element in addition to AR targeted therapy before it goes down the road to differentiation.
so you mentioned about like substitutes, so fibers, stemness,
Yeah.
Have you tried like stemness targeted like therapies?
Yeah, we haven't, but that's obviously there's a lot of interest. So you saw I showed two papers from, a main paper from Charles Sawyer's group and looking at JAK-STAT. So they're very interested in various therapies related to that, to targeting stemness. Yeah. I mean, our approach was related to the epigenetic approach and yeah.
Thank you. I